Hello everyone, I'm a PhD worker at Peking University in Beijing, China. My name is Xuefei Guo. The title of my presentation is Identification and Characterization of Endogenous Retroviruses, also named ARV, upon SARS-CoV-2 infection, which was published 2 months ago in the Journal of Frontier in Immunology. As we all know, the coding gene in our genome only occupies our genome 2%, but the retrotransposer of our genome occupy about half of human genome. And as we can see, ARV belong to the L-tier elements, which occupy 8% of our genome. And the name retrotransposer means the element in our genome could be reversed or transcribed into our genome with the formation of ribonucleoprotein complex, which could enlarge our genome. And we can see that the formation and classification of ARV, as we all know, the ARV consists of several important elements such as GAG, PRO, PO, and ENV. We can see that before millions years ago, the herb retroviruses infect our ancestors, and they could maintain in our genome, and most of them, we can see that this ARV could be divided into 3 families, such as class 1, class 2, and class 3. Class 1 include herb H, herb F, herb W, herb R, herb P, herb E, herb I, herb T. And the class 2 include ARVK, which was composed of HM1 to HM11, and class 3 was composed of herb L. And we also know that the ARV was mutated because which was harm to our genome, but at some conditions such as external signaling or host signaling, such as chemical substances, physical including agents, exogenous viral infection, which may be upregulated the ARV expression in our genome. And also the ARV was regulated by host effect, such as non-coding RNA and functional peptide or protein. In most conditions, the ARV was silenced by our genome and epigenetics modification, but in some tumor cells, the ARV was upregulated, and which may transcribe double-stranded RNA, linker RNA, or mRNA, such as NP9, ENV, REC. However, whether SARS-CoV-2, which result in the break of COVID-19, whether SARS-CoV-2 could induce the expression of ARV is still unclear. In our study, we wanted to know whether SARS-CoV-2 could induce the expression of ARV. So we reanalysis the public data from GEO, which was composed of human RNA-seq data and mouse and monkey RNA-seq data from the GEO public data. Then we conduct comprehensive and system analysis of coding gene and T, including ERVs. And then we did differential express the analysis and functional annotation. As we can see, the heat map from the 34 human sample, we could see that there are some DGs were upregulated upon SARS-CoV-2, and we performed the DG analysis. We could see that there have more DGs in the severe patients compared to mild patients, and we performed the enrichment analysis of SARS-CoV-2. We could find that the PGG analysis of mild patients was enrichment to system lupus eris neural extracellular trap formation, viral carcinogenesis, and the KGG analysis of the severe patients was enrichment to was similar to the mild group. And the geo analysis of mouse and KG analysis was more classical, such as inflammatory response, immune system response, and response to viruses. Also, the KGG enrichment analysis of mouse also enrichment in the NF-κB signaling pathway, PI3K AKT signaling pathway, and TNF signaling pathway, which was consistent with the clinical patients in the hospital. And another interesting thing is that we could see that we use T transcriptor to bioinformatics tools designed to predict the expression and count the expression of ERV and T. We could see that in the mild and severe group, they both have some retrotransposer and DNA transposer were upregulated. For example, we could see that some T and ERV were upregulated in the COVID-19 patients, and we could see that there were some most upregulated T, especially in the severe group. And we also found the transcriptose, including ERV, was upregulated in monkey and mouse group. Then we wanted to know the classification and composition of the T and ERV. We see that in the mild group and severe group compare, the T was composed of LTR line, SINE, DNA set line, and other group. We could see that the ERV1 in the severe group was higher in than the mild group, and we could also see that there are some ERV was only upregulated in the severe group, and there are also common ERV was upregulated in both severe and mild groups, such as the origin one. and the yellow one more 65B was only upregulated in the mild group. And we also compared the T and ARV expression between monkey and mouse. We could see that the most upregulated T was DNA transposer in the mouse group, and the most upregulated ARV in monkey group was ARV1, then was ARV-L and ARV-L ma LR-LTR gypsy. And there are also some common ARV was upregulated in the orange color, and there are some specific upregulated T in both mouse and monkey. Then we use the enrichment analysis with a algorithm named GREAT. The GREAT algorithm was designed to predict the encoding gene region. We could see that the GREAT analysis of mild T, the enriched term include some cetinolone activity and iron chain receptor complex, and the important is the greater analysis of T in severe group. There was some histone modification pathway such as H3K36 methylation and histone H2A monoubiquitin, and negative regulation of histone H3K4 methylation. Then we also performed the great analysis in monkey and mouse group. We could see that in the monkey group, it was also related to some cytokine pathways such as interleukin-1 receptor binding and protein binding, or lipoprotein metabolic process, negative regulation of GT-PAS activity, and the greatness of T in mouse was some cellular metabolic process. Then we wanted to know the difference between mild and severe group, and I wanted to find the potential marker of severe group. As we could see that before, the severe group have more T or ERV was upregulated than the mild group, and we could see that there indeed have some T only upregulated in severe group, but not a mild group. Then we make a vein plot to compare the T between mild and severe. We found that they have 210 T was most upregulated in severe group, and they are composed of LTR subfamily, line subfamily, DNA, and SINE, and Z-line, and others. We could see that the most upregulated T or ERV was ERV1 subfamily. Then we performed the great analysis of the most upregulated T. We found that the most closest gene to T was related to Tau-like receptor binding and histone transformerase activity and transcription Q repress activity. and T-cell antigen process and parasitic, and histone H3K4 demethylation regulated of T-cell mediated immunity, which indicate the unique upregulated T in severe group may contribute to the severity of COVID-19. Then we used the most upregulated 2 ERV in the severe group. We found that it was indeed upregulated than mild and healthy group, which indicated they may be developed as potential marker. And we of this was predicted by the algorithm. Then we needed to validate the accuracy of prediction. Then we used the two other viruses like HSV and VSV, and we wanted to know whether this algorithm was accuracy. So we prefetched the BMDM from mouse and then infected by VSV and HSV. We found that both VSV and SV could upregulate some T and ERV in the BMDM, such as the ERVB21 and ERVB51 and RIX-C was the top 3 upregulated ERV by HSV, both HSV and VSV. Then we designed the primer to validate these results. We found that ERVB2 have about 80 copy number, and we use bioinformatics tools to validate 4 of its copy number, and we found that the result was consistent with the prediction. So it demonstrated that our prediction was accuracy and robustness. And there are still some important questions needed to explore, such as what's the role of upregulated ERV? Could the host PR can recognize this upregulated ERV? How do they influence the host viral interaction and the regulation of the host immunity? That's all. Thanks for your attention.